Hi everyone, welcome back to my channel, and today we are going to learn about transcription. We have already discussed about central dogma of molecular biology in our previous video. So before watching this video on transcription, I want to recommend you guys my previous video on DNA replication. So let's get into the topic. So transcription is the process where RNA is synthesized from DNA. It involves mainly three steps. First one is initiation. Second one is elongation, and third one is termination. Some steps are different in case of prokaryotic and eukaryotic cells. We will discuss this also. So the first of all, that is initiation step. What we have to know is this is DNA. In case of this transcription, these two strands of DNA, one strand is known as the template strand, and another strand is what we call is a coding strand. In this template strand, there is something known as promoter site. This promoter site is not transcribed. It is the site where these RNA polymerase enzymes are going to act and make RNA. So here there is the enzyme called RNA polymerase. It can form all the forms of RNA except RNA primers. It does not need RNA primers. So this RNA polymerase enzyme are different in prokaryotic and eukaryotic cells. In prokaryotic cells, there is only one RNA polymerase enzyme which can synthesize all forms of RNA. But in case of eukaryotic cells, there are three different types of RNA polymerase. RNA polymerase type 1 will make rRNA, RNA polymerase type 2 will make mRNA, and RNA polymerase 3 will make tRNA and 5S rRNA. Now, this RNA polymerase enzyme will also need transcription factor. The addition of these proteins are required to initiate the transcription. So these are different in case of prokaryotic and eukaryotic cells. In prokaryotic cells, the transcription factor is called the sigma factor. And in eukaryotic cells, there is the transcription factor D, transcription factor B, and transcription factor E. Now in this initiation phase, there is the binding of RNA polymerase to the promoter region of DNA, and with the help of this transcription unit also, it will initiate the formation of RNA. Now the promoter region. These promoter regions are also different in case of eukaryotic cells and prokaryotic cells. In case of prokaryotic cells, the initiation factor is known as Pribnow box or -10 region. Another is -35 sequence or -35 region. What does it means is, if there is a, this is the promoter site, if transcription will start from this point, so there are 10 base pairs here, so the name is given as -10 region. Likewise, if there are 35 sequences of nucleotide, then it is known as 35 sequence. In case of eukaryotic cells, the promoter regions are Tata box. It is very common in eukaryotic, it is a most common eukaryotic cell promoter, and it will bind to the transcriptional factor D. Likewise, another is cat box, and the third one is GC box. So these are the promoter regions where our RNA polymerase enzyme and transcriptional factor are going to act. So this is DNA 3' to 5', 5' to 3'. First of all, there should be unwinding of the DNA. So this unwinding of DNA will be done by this RNA polymerase and transcriptional factor. These both are going to unwind DNA. So this is our RNA polymerase enzyme which is unwind the DNA. So this is the template strand and this was our coding strand. So RNA is going to form in the direction 5' to 3' direction. So here the process of elongation starts. So this is how our RNA is going to elongate till termination. So there is termination in prokaryotes which is of two different types, that is row dependent termination and row independent termination. In case of eukaryotic cells it can terminate at some random points, but in case of prokaryotic cells the termination can be of two types. One is row dependent and another is row independent termination. First of all, row dependent termination. So there is a protein known as row protein which has two activity. One is the helicase activity and another is the ATPase activity. This helicase activity is it will separate the newly synthesized DNA from RNA. So this RNA will get separated from DNA. And ATPase enzyme, it insert into the transcriptional bubble. So this is our transcriptional bubble. So it will insert itself in transcriptional bubble and interfere the movement of RNA polymerase. As this RNA polymerase was making RNA, it will interfere its function so that DNA can be separated from the RNA can be separated from the DNA. So another is row independent termination. Now let's discuss about the differences between the prokaryotic and eukaryotic transcriptions. So the first difference is of promoter's region. We already discussed the promoter regions in prokaryotic cells will be Pribnow box or the -35 region. Likewise in eukaryotic the promoter regions are Tata box and Cat box or maybe the GC box. In prokaryotic there is single RNA polymerase, and in case of eukaryotic cells there are three different types of RNA polymerase. Now in case of transcriptional factor there is sigma factor in case of prokaryotic cells, and in case of eukaryotic cells there is transcriptional factor D, transcriptional factor E and transcriptional factor B. In case of eukaryotic cells there are involvement of enhancers and silencers. These enhancers are increasing the expression of gene or they are activating the transcription process, and the silencer are decreasing the expression of gene of the chromosome or they are just silencing or repressing the transcriptional factor. Another very important, there is no any enhancers and silencers in prokaryotic cells. The fifth difference is there is no splicing of primary transcript. We will discuss it later. But in case of our eukaryotic cells, there is involvement of splicing. So the last difference here is transcription process and translation process occurs simultaneously in case of eukaryotic cells. So this transcription and translation occurs simultaneously. But in case of eukaryotic cell, it cannot occur simultaneously. The process can't occur simultaneously. Now, let's go in very important topic, that is the post-transcriptional modification in eukaryotic cells. So, when we read about this central dogma of molecular biology, we know when RNA is synthesized from DNA, we called it as a transcription, and when proteins are synthesized from RNA, it is known as translation. But it can be modified in case of prokaryotic and eukaryotic cells. In eukaryotic cells, there occurs some post-transcriptional modification. So, it can be called as if RNA is synthesized from DNA, it is called transcription. But if this hnRNA is synthesized, that is called as transcription. And whenever there occur this splicing, only this hnRNA can be converted into mRNA, and later this will convert it into protein with the help of the process translation. But in case of prokaryotic, it is very simple. The DNA is formed and RNA can be synthesized from DNA with the process of transcription, and again this mRNA will be converted into our protein with the process of translation. This specific process or step of forming of mRNA from hnRNA is splicing, or it can be called as the post-transcriptional modification in eukaryotic cells that only occur in case of eukaryotic cells. So in this post-transcriptional modification of eukaryotic cells, let's first know the initial transcript is called the hnRNA, which is the heterogeneous nuclear RNA. It should be converted or modified into mRNA, so it is also known as pre-mRNA. So this is the structure of hnRNA. It have 5' to 3' direction, and in between there are exons and introns which are alternative. So these exons, they contain the actual genetic information. For coding for protein and functional RNA, but these introns, they do not code for protein, but they are important for regulation of gene expression. So these introns will be inside the nucleus, and these exons will exit the nucleus, and this will be expressed. Now, in case of this post-transcriptional modification, there occurs mainly 3 steps. First one is 5' capping. Second is addition of poly-A tail. And third one is splicing. So this 5' cap occurs in this 5' site. Addition of poly-A tail will occur in this 3' site, and splicing will occur in these exons and introns. So let's learn step by step. First one is the 5' capping. Here the heterogeneous nuclear RNA have this 5' to 3' direction. So what happens in 5' capping is a cap of 7-methylguanosine will be attached to the 5' end of our mRNA. So there is addition of guanosine, and there is addition of methylation of this guanine. So this occurs due to the help of this enzyme methyltransferase. The enzyme methyltransferase that is present in the nucleus will add guanosine to this 5' structure, and this methyltransferase which is present in the cytosol will add this methyl group to our guanosine. So what is the function of this capping? So the function of capping is to facilitate the initiation of translation and stabilize the RNA. Another is addition of poly-A tail. So this polyadenylation signal sequence is that there will be addition of around 40 to 200 adenine molecule. These nucleotides will be added in the 3' end of our mRNA. So it is called as a poly-A tail. So here there will be the 5 cap and there will be a tail where there will be a lot of adenine. So the function of this polyadenylation is also to stabilize the mRNA and facilitation of exit of mRNA from nucleus to cytosol. Third one is splicing. So splicing is removal of our introns, because we have already discussed these introns do not code for proteins. So removal of introns from hRNA. So this will help to rejoin the exons only. Let's discuss the process of splicing. So in this splicing, what happens is the primary transcript combines with snRNPs or SNRPs, which is also called as small nuclear ribonucleoproteins. So this both will combine and form the spliceosomes. So what happens is that loops of mRNA is formed with the introns. So this is our exon and this is next exon. In between there is intron. So in the 5' side there is GU nucleotide and in 3' there is AG nucleotide. So this spliceosome will come and it will cause these introns to loop around. So like this it will loop around and this looping part is known as the larynx. Now this larynx will release and it will remove the introns so that exon will join with another exon. So now this exon can be come out of from the nucleus to the cytosol and our mRNA is formed. So this is all for today. I hope you guys enjoyed. We will meet up in our next video.